Hi there, I'm Mrs. Gay, and I'm starting on page 52, Chapter 4, Section 1. This section is about microscopes, and quite honestly, all you really need to do is be familiar with the fact that there are different types of microscopes, and we will work largely with a light microscope. And so think about the onion root cell. You're never going to get asked to name the parts of a microscope. You're never going to get asked how to focus a microscope. But you should be aware that many of the things that we study in biology are smaller than we can see with the naked eye, and that the microscope is a valuable tool to help us see things that we normally couldn't see. So we have the light microscope. We have an electron microscope, scanning and transmission. So a scanning electron microscope shows us the surface of things, and while a transmission microscope actually puts energy through the object and we can actually see inside. Okay? That's plenty to know. The one really important thing in this whole section is at the first paragraph in the second column of page 52. Okay? And it's probably not a huge deal, but Robert Hooke discovered cells. Cells, and this is actually total trivia, cells were not, don't think prison cells, but cells were small rectangular quarters that were occupied by monks in monasteries. And that's what they reminded him of. And so that's why he called them cells. Okay? And so that's a little bit different. But the one piece that's in here listed is cell theory. Okay? And we'll go back and revisit cell theory in another section in this book too. But the big points about cell theory is that the cell is the basic unit of all living things. That's sort of one facet of the cell theory. Okay? The second big idea about the cell theory is that all cells come from pre-existing cells. Okay? And we'll see in a later section that these ideas came first from Virchow and then later from Schleiden and Schwann.
Do you want?
I know, they were both German.
Do you want the name, like, would a question on the test be, what are the names of the?
No, they would not. They would not ask you who discovered, who were the major contributors to cell theory and have a whole bunch of names. But they might mention Schleiden and Schwann in association with cell theory. And so I mention it because I want you to, if you see it, be, oh, right. You may not be able to pull it out of your hat, but if you saw it, you wouldn't freak out. Like, oh, my God, I have no idea who these people are or what they did, when really it's not that big a deal. Okay?
I would say, like, the theory formed by
Schleiden and Schwann. Or if you think about it, okay, so if you think about it on the free response question for the cell membrane essay that I asked you to write, and it was talking about the fluid mosaic model, and it was talking about the Singer model versus the Danielson model, or Danieli, I can't even remember who the other person was, model. If you knew who had come up with that idea and you listed it, you might get a point. But there were plenty of other ways to get your 10 as well. So if you're good with names and you like sort of historical things, great, remember the names. But it's not going to hurt you or necessarily help you a whole lot either way. But I tell you them mostly so that you would be familiar with them and not be surprised if you saw them. Does that make sense? Yeah. So I would not, I would look over this section, but I would not spend an extraordinarily large amount of time on that section. So we're going to move on to section 4.2, which is a big deal. Okay? Most cells are microscopic, and if you look at figure 4.2a on page 54, you can see what we can see with the unaided eye. You can see that that overlaps with what we can see with the light microscope, which also overlaps with what we can see with an electron microscope. Okay? So you should have a sense of size. Okay? Notice this, though, that's interesting. We'll come back to this later. A chicken egg. Notice how big it is. Ostrich eggs, emu eggs, even bigger. Frog eggs, smaller. But notice most plant and animal cells, the nucleus is actually bigger than most bacteria. A mitochondrion is smaller than most bacteria, but not a lot smaller. And that's a preview of endosymbiotic hypothesis, how we got from prokaryotic to eukaryotic. Notice that viruses are even smaller. Ribosomes are even smaller than a virus. And then we get down to the macromolecules and smaller. So just having a small sense of the scale of things. The other really important idea in this chapter is the idea of surface area to volume. It's all about surface area. Okay? And the reason that it's all about surface area, as things get bigger, the surface area only increases as the square, where volume increases as a cube. Okay? And at the top in figure 4.2b, they give you a really nice illustration of what happens where the volume remains the same, but they break it up into smaller cubes and we drastically increase the surface area. We find that happening all the time. Think about your brain. Think about your intestines. Think about your lungs. Everything is highly folded and convoluted to give greater surface area.
And what's the benefit of having that?
Well, the benefit of having a large surface area, think about exchange of nutrients. And think about your osmosis diffusion lab with those agar blocks. They started out pink. We put them in hydrochloric acid. They turned clear. Which cube took the longest time to get clear? The big one. Now think about this. I could have had a cube that had the same volume as the biggest cube, but what if I would have made it flat and long and thin? It would have taken way less time because we would have exposed much more surface area than by keeping it in its nice sort of handy cube shape. Does that make sense? And so surface area to volume matters at a cellular level because we have to get waste out and we have to get nutrients in. And so if you think about nutrients diffusing into the cell, that really big cell that stayed pink in the center, nutrients never got into the center of the cell. Waste in the center of the cell still would be inside the cell. And that would end badly for life. So it's about exchange with the environment.
Are there like cells that have like greater, like just an example of an actual cell that has better surface area to volume than other cells do? Like,
sure. Think about a nerve cell that would be really long and thin. That's going to have a much greater surface area than, say, a cuboidal cell.
So just overall thickness.
So, right, so thinness and stretching it out. Think about a roll of aluminum foil. You could take a roll of aluminum foil and you could wad it up into a cube. Okay? And you'd have a cube of aluminum versus rolling it out super thin. It's going to have much more surface area all rolled out thin. But what do we have to do with the aluminum foil all rolled out thin? That would be really tough to store unrolled.
Yeah.
So what do? We fold it up, we roll it up, we compress it. And so if you think about the internal structure of much of you, you're folded and convoluted. Okay? So I'm going to stop there. We'll start with Memo 2, looking at Section 4.3.